Dear students, after studying this module, you shall be able to know about the molecular structure of ion channels and transportation of material from the external to across the membrane, functioning of physiological processes including nerve impulses, muscle contraction, regulation of cell volume and cell signaling in all organisms, mediating materials for ion transportation, different mechanistic for selective ion transportation. The concepts of cellular electricity and ion permeable pores had broadly developed in last 3 decades, but physiologists postulated the molecular pores in membranes several centuries ago. The history tells that the process of osmosis was discovered by Jean Antony Nollet in late 1700s. Later, in 1843, Ernst Walk-Bruch postulated pores, canal to account for the osmosis process and postulated pores no more than few water molecules wide. He imagined capillary canals in porous membrane. Soon, Carl Ludwig in 1852 to 1856 adopted Bruch's pore theory to explain the ultrafiltration in the kidney glomerules, and Adolf Fick in 1855 devoted much space to discussing diffusion of salt and water in molecular pores of membranes in his discussion proposing Fick's first and the second laws of diffusion. Such important concepts were discussed by these physiologists before the size or even the clear existence of molecules was known, and before the word membrane connoted anything but some multicellular sheet like epithelium or even the sheets of parchment. Nevertheless, from 1850 onwards, mechanistic books of physiology would discuss the possibility of semi-permeability and membrane pores in an early section on general physiology as an explanation for osmosis and filtration. German books used the word canal and English books said pore. In each generation, various new investigators propounded and were given credit for the pore theory. For example, in 1925, Michaelis proposed that the permeation of small ions might be controlled both by the charge on the pore and by the pore diameter relative to the hydrated ions by his experiments on artificial colloidal membranes. Ion channels function to orchestrate an exquisite array of physiological processes, including nerve impulses, muscle contraction, regulation of cell volume, and cell signaling in all organisms. The electric current in a signaling event is generated by ion flux across the cell membrane, that is controlled by an opening and closing of the ion channels, including those permeable to potassium, sodium, calcium and chloride ions. The direction of the ion fluxes is determined by the membrane potential and preset transmembrane ionic gradients, which are established and maintained by specific ion channels and the sodium-potassium ratio ATPase. For certain signaling modes, such as the generation of the action potential, it is important that the ion channels involved are only permeable to specific ions and impermeable to the others. Particularly important to signaling in nerve and muscle are potassium and sodium channels, which display a very high level of selectivity. Therefore, some ion channels have evolved to exhibit high ionic selectivity, which is fundamental to electrical signaling. The transport mechanism must be highly specific in favor of one water-soluble chemical species. It must be able to move that species from one aqueous region to another through a bilayer of variable polarity with a hydrophobic core and it must do this fast enough to respond to rapidly changing inter and intracellular conditions. The complex nature of the process has been called supramolecular biology by some to reflect the many non-covalent interactions and play between cell membrane, the transmembrane transporter, whether ionophore, aggregate or channel forming protein, and the transported species. Molecular structure of ion channels. The ion channel are the component of lipid bilayers of cell membrane, as can be seen from the figure. A cell membrane consists of hydrophilic, which is water soluble phosphate head groups attached to a long lipid fatty tail, and is thus an example of amphiphile. In a body's aqueous environment, the hydrophilic head groups are attracted to the surrounding medium, for example, with the hydrogen bonding and the dipolar interactions, while the organic tail is repelled. This results in a bilayer arrangement in which the organic components are all hidden away from the solvent while the hydrophilic portions face out. The phospholipid layers held together by interdigitation of their hydrophilic alkyl tails and form a lamellar structure with their polar internal and external surfaces separated by a non-polar region. In their extended form, each phospholipid is about 3 nanometer in length. However, when the hydrophobic tails of two lipid monolayer intertwine, a bilayer 4 to 5 nanometer thick results. The membrane is also host to large biomolecules with particular functions that can bind to or in some cases penetrate its surfaces. The bilayer's fluid mosaic structure allows it to reorganize when incorporating the macromolecules, but remains intact unless seriously compromised. Anything that is to pass through the cell wall must therefore be able to pass this lipophilic fat soluble region. Although the hydrophobic region forms an effective barrier between the extra and intracellular environments, but it can still be penetrated by a number of biological molecules such as proteins and synthetic compounds designed to insert through the polar exterior to the non-polar interior of the bilayer. Challenges is to use this phenomena of transport specific ions through this apparently impermeable barrier. Active transmembrane transport occurs in two ways: migration of facilitated by species, specific complexing agents that shuttle through the bilayer, and channels formed by the transmembrane protein and similar molecules. Simple migration occurs when a carrier or host molecule traps a guest ion pair or a molecule and transports it across the cell membrane. To function effectively, the host must be selective for only one guest species. The host needs to be water soluble to bind the guest in solution, but complex has to enter the polar region of the bilayer, then across the non-polar core before the process is reversed for the guest to be released on the other side of the membrane. The host must have polarizable regions to bind a guest or coordinate to water, and the ability to change conformation, inverting these regions to encapsulate the guest and present a lipophilic exterior when passing through the hydrophobic region of the layer. There are many naturally occurring highly selective carriers, and most are able to transport up to 10 to the power of 4 guests per second. Ionophore mediated transport. Organic ligands that bind to alkali metal and alkaline earth cations are known as ionophores, literally an ion carrying compounds. Valinomycin, a cyclic natural antibiotic, consists of an alternating D and L valine separated by dehydroxyisovalerate and L-lactate groups as shown in the figure, binds to potassium ions 1000 times better than the sodium ions and exemplifies carrier facilitated transport. The free ionophore crystallizes as a rectangular box, roughly 1.7 nanometer long, 0.9 nanometer wide and 1.0 nanometer high, held in place by the intramolecular interactions. The complex has a tubular shape, 1.0 nanometer high with the internal cavity of 0.4 nanometers, containing the metal ion and an overall diameter of 1.5 nanometer. 6 carbonyl oxygen atoms bind the metal in an octahedral environment, forcing valine and isovalerate side chains to form a hydrophobic exterior to the tubular complex. It is the inversion that occurs upon metal binding that turns the hydrophilic ligand into the hydrophobic lipid-soluble complex. Binding constants for a range of naturally occurring ionophores are given here in the table. Note that many ionophores bind strongly to potassium ions while only monicin actually binds sodium ions within selectivity. Siderophore mediated transport. Yeast, fungi and bacteria such as Salmonella genus and Escherichia coli need to sequester iron from aqueous solution. Unfortunately, water-soluble ions is easily oxidized to ferric ions in air, which makes it much less soluble. At concentrations of about 10 to the power of minus 17 molar for free ferric ions in aqueous solution, it is essential that every available ion is bound effectively and transported across the cell membrane. This is achieved by siderophores, iron-specific chelating agents that bind ferric ions in an octahedral pocket, usually comprising 3 bidentate catechol or hydroxamate groups as illustrated in figure. Binding is specific and very strong with binding constants ranging from 1020 to 1050. The resulting complex is targeted by a membrane-bound receptors to return it to the bacterium. Once inside the protection of the cell-localized conditions, reduce ferric ions back to ferrous ions, which is then released as the siderophores. Affinities for the reduced form are much lower than that for the oxidized form. Anion transport. Anions can also be transported across membranes. In nature, chloride is transported by proteagiosins, which comprise 3 conjugated pyrrole groups and a lipophilic alkyl tail as shown in figure. These simple compounds simultaneously transport chloride ions and the protons by what is termed as symport mechanism. As it is neutral, it can traverse the phospholipid bilayer with much greater ease than the charged complex. Cation channels. Cations come in many shapes and sizes. The simplest is the lone proton, which may jump from base to base along a small channel. Then there is inorganic ions with no directional preferences for bonding, such as the alkali or the alkaline metals, and the ammonium ions, which is a tetrahedral but appears spherical when hydrated. At the other end of the spectrum of the structural complexity, we have organic cations and hydrated transition metal complexes with non-uniform charge densities. 4 metal cations, sodium, potassium, magnesium and calcium, are of primary importance. Sodium and potassium are ubiquitous in biological systems where they are employed to control membrane potential as well as intracellular levels of hydration. Several types of sodium and potassium transporting channels are known to exist, and some, notably the inwardly rectifying potassium channel KSCA, isolated from Streptomyces lividans, the filter region of which is illustrated in the figure, have been subject of intense investigation by multiple techniques. Sodium channels. Voltage gated sodium channels usually remain in the closed state but open in response to minute voltage changes. The techniques of channel cloning and site directed mutagenesis have allowed the sodium permeability of several mutant channels to be probed. This is done by analyzing the responses of the channels to site-specific anesthetic molecules and toxins. Tetrodotoxin, isolated from pufferfish, binds to particular domains found near the outer mouths of sodium channels and causes channel failure. The response to tetrodotoxin can therefore be used to determine the presence of a specific protein sequence in a channel. Certain scorpion toxins can also be used for this purpose. Voltage gated channels activated by changes in membrane potential leading to conformational changes allow the channel mouth to open to accommodate appropriately sized ions, but once they enter the channel protein, these ions face a selectivity filter. Potassium channels. There are two main groups of transmembrane potassium channels. The first includes protein with 6 transmembrane domains such as voltage gated potassium (Kv) channels and the calcium activated potassium (Kca) channel. The second includes the inwardly rectifying K+ Kir channels with 2 transmembrane domains. Kv channels are hetero or homotetramers according to electron microscopy and mass determinations, implying that the ions are channeled through the pore formed at the confluence of the proteins. Scorpion venom inactivates the outer mouths of the Kv channels by binding in an analogous manner to tetrodotoxin in voltage gated sodium channels. The selectivity filter appears to about 0.5 nanometers from the extracellular phase of the protein, and mutagenesis experiments have shown that the removing a tyrosine glycine dipeptide sequence allows other alkali metals to pass at the rates similar to potassium ions. Kca channels require a particular calcium concentration to initiate potassium conductance, though a little is known about their structures. Anion channels. As with the neutral and the cation channels, anion channel forming proteins are found throughout the nature. In 2002, the first high resolution 0.3 nanometer X-ray structures of the anion channels were published by McKinnon Group. Although they are from the CLC acronym, CLC stands for chloride channel family. These proteins conduct bromide as well as chloride, though the former is less common in the physiological environment. CLC channels from Escherichia coli, a Salmonella typhimurium, illustrated in figures, comprise with 2 subunits in an anti-parallel manner. Each subunit contains 18 alpha helices arranged in a complex topology in which the nitrogen and the carbon terminal parts of each subunit are organized in an anti-parallel architecture that creates a pseudo two-fold symmetry axis within each monomer. Crystallographic structures of EC-CIC and ST-CIC have been shown here in the given figures. For each protein, The two subunits are indicated by different colors. The permeation pathway can be inferred from the position of the two chloride ions in the magenta color. The helices interact with each other to leave a central pore, and two of these channels align also in an anti-parallel fashion to give a structure with two channels. Anions pass through a selectivity filter comprising isoleucine, phenylalanine, serine and tyrosine, though interestingly there appears to be no strong interaction with the positively charged residues. It is the hydrophobic regions that induce anions into the filter. A further feature of the channel is the presence of a conserved proline containing sequence of amino acids at the end termini of the alpha helices that give the channel a twist. As noted, above the proline, twist motif has been exploited in the synthesis of artificial anion channels. To summarize, in this module, we have learned that ion channels are ion permeable pores which are component of lipid bilayer of the cell membrane. Ion channels function to orchestrate an exquisite array of physiological processes including nerve impulses, muscle contraction, regulation of cell volume and cell signaling in all the organisms. The selective transportation of ions by many naturally occurring highly selective carriers such as ionophores and siderophores, which are most able to transport up to 10 to the power of 4 gas per second.